Hi, everyone. Welcome back to the DBMI Seminar. Today's speaker is Dr. Manuel Rivas. Dr. Rivas is an assistant professor in the Department of Biomedical Data Science at Stanford University in Stanford, California. He has a Bachelor of Science in Mathematics from MIT and a Doctor of Philosophy in Human Genetics from the Nuffield Department of Clinical Medicine at Oxford University, where he was a Clarendon Scholar. He also did additional training at the Broad Institute in Cambridge, where he led the Helmsley Inflammatory Bowel Disease Exome Sequencing Program to understand the genetic factors that contribute to ulcerative colitis and Crohn's disease risk. So with that, I'll hand it off to our speaker. Thanks for being with us today.
Hello. So thank you very much for the kind introduction. As Yes mentioned, I'm faculty in the Department of Biomedical Data Science at Stanford. And today it's a real pleasure to be with you to share with you some of the research that we've been doing in my lab, specifically focused on the genetics of population cohorts across the world. And the focus today will be on the application of some of the methods and some of the techniques that we've developed in the lab, focused on human genetic studies to 35 serum and urine biomarkers from the UK Biobank cohort. So just an intro slide that will describe a bit about the UK Biobank for those who are not familiar with it. So UK Biobank is a population-based cohort from England that makes available data to any bona fide researcher around the world. Right now, its makeup is of approximately 500,000 individuals, where health records, verbal-based questionnaire, and web-based questionnaire data on approximately 500,000 individuals is available. Genotyping or genetic data is available for 500,000 individuals as well. Right now, there was a recent data release of about 200,000 exome sequencing data set that is probably the largest single point exome sequencing data set around the world. Furthermore, in the study that I will share with you today, it will be the combination of the genetic, the health record data, and the baseline biochemistry data from 500,000 participants that was also made available. And for those that may be interested as well, there's an imaging data set with plans of about 100,000 individuals, with the current data release being of 40,000 individuals where they have brain imaging, chest, abdomen imaging available. Okay, so as far as the research study is concerned that I will share with you today, from a human genetics point of view, one of the key questions that we wanted to address from combining the human genetic data that was made available from UK Biobank with the biomarker data that was also made available and the health record information data that was available was to address three questions. One was, can we identify strong effect genetic variants that may pinpoint new genes that may be worthwhile pursuing either as a therapeutic hypothesis? Two, could we possibly find map these common genetic variant associations that are commonly identified through these genome-wide association studies? And third, could we develop predictive models from the genetic data, taking advantage of the property that many of these traits will have many genetic variants across the genome that have some non-zero effect to it, these so-called polygenic risk models. And what I'll try to do for the talk is, for those that are not familiar with the genetics, human genetics terminology, I'll give some background as to what they mean and why is it that the question is important as far as a human geneticist point of view. And second, from a biomedical informatics point of view, why integrating these data sets is at a very interesting moment in time. Now, to credit both graduate student, PhD student researchers that led this work, along with the team that we collaborated with. Those two graduate student researchers include Nasa Sino Armstrong. He's a PhD student at Stanford University, and Yosuke Tanigawa, who is also a PhD student in the biomedical informatics graduate program at Stanford University. So just to give you a background on the actual study design, here what we did was to combine data from 363,000 individuals, unrelated individuals that had serum and urine labs, 35 serum and urine labs measured. In addition to that, we used some HLA allele type information data that was available to us and some copy number variant calls that was available to us as well, in combination with the genome array and the mutation data that UK Biobank made available. The study itself was just published last week in Nature Genetics. So if any of you want to read about the study in detail, you can read the paper along with the supplementary material. And there's a ReadCube link that I'm happy to share as well. Okay, so one of the first things that we did with the data was to combine the serum and urine labs with the genetic data and apply the typical association analysis that you perform in human genetic studies. These are the GWAS or genome-wide association studies applications where you go variant by variant and apply either a linear regression or a single logistic regression at a time independent of each other. And as you can see from the results, at least, there were many genetic variants that exceeded a p-value threshold of 5 times 10 to minus 9 that we used for this study. Furthermore, we used methods that are able to estimate the heritability of each of the biomarkers. And for each of the biomarkers, we estimated the heritability. And for each genetic variant that was identified, we asked the question, can we identify other phenotypes that may be associated with those genetic variants as well that are clinically relevant phenotypes in a PHI-WAS-like study design? So you go from identifying the association to asking the question, is that variant also associated with any other clinical outcome data that we have access to? So this is a typical workflow, I think, for many genetic studies nowadays. There are methods that estimate heritability of a trait, and some of those methods actually we ended up stumbling into some of the limitations through this study design applied to these data. And one was, what happens in the context where there is a heritable trait, but most of the heritability is explained by a handful of variants, if not, just one specific loci. And that happens to reach into some of the limitations of the assumptions that are made by some of the methods that have been implemented to date. One example of that is lipoprotein little a. Okay, furthermore, one of the key questions by combining these data sets, we asked, are any of the biomarkers causally associated or causally related to any disease outcomes that we've studied? We used an approach referred to as Mendelian randomization for assessing those relationships. And in addition to that, we trained polygenic risk score models for each of the biomarkers themselves. And in combination, we realized that by combining the polygenic risk scores of the biomarkers in combination with the disease outcome polygenic risk score, we could possibly improve the predictive performance of the polygenic risk score to predict disease outcome. And I'll share with you some of that intuition later, but there are some very interesting upper bounds as to where that performance can reach. So just to give you a summary as to the number of LD independent associations that were identified in this study, we are at a moment in time where having genetic data combined with a trait on hundreds of thousands of individuals is accessible to almost anybody. And here, one of the cool things is that we are able to reach power to detect associations in almost every common genetic variant across the genome. And here in this study design, at least, we're able to identify hundreds of LD independent associations for many of the cardiovascular, liver, and even renal biomarkers that were studied in this research project. So here is a representation of those genetic associations in the form of what Masahiro Kanai from the Broad Institute. He was in Japan when he developed this plot. And here what this plot does is to summarize the number of LD independent associations and the number of associations for each of the biomarkers across the genome. So here we have the 35 biomarkers for each row or circus. Masahiro referred to this plot as the Fuji plot, which is just fantastic and extremely relevant, given that it was first applied to Biobank Japan, and here we apply it to UK Biobank as well. So this work was led by Yosuke Tanigawa, and one of the additions that we added to this plot or representation of the association results was this concept that we were focused or zoom in on genetic variants with strong effects on the outside circus. And the idea would be to pinpoint coding variants specifically. One of the nice things about the UK Biobank is that its array design actually consisted of tens of thousands of coding variants with a minor allele frequency threshold, lower bound threshold of about 1 in 10,000, 1 in 100,000. So in this study design, we did have pretty good power to detect some of those genetic variant association signals. And for each coding variant, what we did was to label them, especially if they had an absolute effect size greater than 0.1. So if you were to plot the distribution of effect sizes estimated from GWAS studies from common variants, most have the property that they would lie between negative 0.1 and 0.1. In fact, it's probably about 95% of associations fall within that range. And one of the beautiful things about having access to rare coding variants specifically was the ability to pinpoint rare coding alleles that had strong effects and reached genome-wide significant levels as well. And just to walk you through to some of these genetic variance associations, what I've done in the next set of slides is to go through some of the genes that are indicated by these analyses to be directly relevant to one of the categories of biomarker levels. So as I mentioned, we studied 35 serum and urine biomarkers. A set of those biomarkers fall into a category of being bone and joint biomarkers that consist of alkaline phosphatase, calcium, and vitamin D. In this study, we identified coding variants in genes like GPL-D1, ALPL, and one of the calcium receptors to be associated with one of the levels of these three biomarkers. And all three had variants with strong effects. For cardiovascular biomarkers that we studied in the set of biomarkers that were analyzed, that includes ApoA, ApoB, cholesterol, C-reactive protein, HDL cholesterol, LDL cholesterol, lipoprotein, and triglycerides. And there we identified multiple rare coding variants in genes like PD3B, ANGPTL8, ACACB, PLA2G12A, CPT1A, and PCSK6. I think many of you may have heard of PCSK9. One of the cool things about this study is that it also identifies other genes like PCSK6 and ACACB to be associated with cardiovascular biomarkers. For diabetes, the diabetes biomarkers that were assayed in UK Biobank include glucose and HbA1c. And here we have coding variants in 4 genes that are directly implicated with these biomarker levels that I've highlighted here. For hormone levels, that includes IGF-1, sex, SHBG, and testosterone levels. And here you see 3 genes with coding variants with strong effects in hepatocyte growth fact-like genes. So here we have HGFAC, HGF, and HNF4 alpha, all with rare coding variants with strong effects on hormone biomarker levels. For liver, the liver biomarkers that we're studying include alanine aminotransferase, albumin, aspartate, aminotransferase, bilirubin, GGT. And here you can see a set of genes, including genes like SERPINA1, SLC3810, with coding variants with strong effects that on these liver biomarkers. And last, in the set of biomarkers, I think that were especially well-powered in this study relative to what was available in GWAS domain for rare coding variants include the renal biomarkers. In the UK Biobank, they measured creatinine levels, cystatin C, microalbumin in urine, non-albumin protein, urate, urea, phosphate, and potassium and sodium in urine. And here, many genes, including SLC transporters, that we identify rare coding variants to have strong effects on one of these renal biomarkers. So all in all, I think this summarizes a key set of genes with rare variants with strong effects that directly pinpoint these genes as being directly associated with the biomarkers that we studied. So one of the other activities that we did apply was fine mapping of the GWAS associations. This work was led by Christian Benner, who developed FineMap. And what we did was to combine the GWAS summary statistic data with the LD reference panel from the UK Biobank. And then at the end, what we ended up getting was to turn the problem around and ask the question, do any of the variants that are associated have enough statistical evidence, at least, of being the one driving the association signal in a specific region? And at the end of the day, you get a posterior probability of the variant being a causal variant, along with the number of other variants that are in the credible set as well. So nonetheless, all this work is in silico. So I think that there's a huge caveat still in this work, in the sense that I think a lot of these genetic variants that are fine mapped need to be integrated with functional follow-up as well. Same thing with the rare variants, but with the rare variants, it's a bit easy to tease apart the LD the patterns. But here with common variants, and just to give you a summary, here we find that many of the regions that we have signals for, or GWAS signals for, have multiple variants or multiple distinct signals per region. Here, for example, we have 33 regions that are fine mapped with only a single signal. Many of the other regions that have been fine mapped have at least two signals associated with it. Approximately 2,500 signals were fine mapped to a single variant. So I think this gives a good set of candidate variants to move forward with in terms of thinking about functional follow-up for these biomarkers. And just to give you a summary of the fine mapping that was inferred from the FineMap program, there's a broad range of fine map variants based on posterior probability of the variant being a causal variant. And here you could see that, for example, for the cardiovascular biomarkers, we have tens, if not hundreds, of variants that are fine mapped to a single variant with posterior probability greater than 0.95. Just to give you an example of the fine mapping from common variant associations, here is two sets of missense alleles with pleiotropic effect in HGFAC. Here you can see that genetic variants, there's a 1% and 34% monoallele frequency coding variant that have quite pleiotropic effects across the 35 biomarkers that we were able to obtain data from UK Biobank. And with the strongest effect being on the sex hormone SHBG. So for the third component of the talk, what I do want to do is to walk you through some of the polygenic risk models. And please, if you have questions, please feel free to interrupt and ask questions in the meantime. We're hoping that this is as interactive as possible. If there's any questions that you'd like to clarify before moving into the polygenic risk score models. Would love to clarify with everybody. Okay, so with that, so in terms of the way genetic studies are done, we have two views of the world for running a genetic study. One is the typical GWAS univariate model that we're used to. Now, the idea there is that for each genetic variant that you're trying to assess association for, you run the model independently of each other. So you run one logistic regression or one linear regression, fit with the covariates that you're interested in. You do a hypothesis test of the least squares regressions that you ended up applying, and you get, at the end of the day, a p-value, estimated effect size, and some 95% confidence interval for the genetic variant that was tested. So this provides a bit more of the direct interpretation. The computation is quite convenient, and it typically works well with small sample sizes. Nonetheless, if you were to use it as a prediction algorithm, it would have weak properties. One of the reasons is that what happens in genetic studies is that for the sets of variants that are tested, usually you have that genetic variant coming along with many other variants that are highly correlated with it, so-called LD friends. In fact, if you were to consider any common genetic variant, there would be at least a handful, if not tens, of genetic variants that have R squared of equal to 1. So this initially has helped a lot of the human genetic mapping activities in terms of being able to go from building out a genetic variant map, building out then the list of markers that tag the rest of the markers or genetic variants that are in the genome that have been identified, and then using a subset of genetic variants to then be able to apply a GWAS style approach across the genome, while maintaining good coverage of the genome for common variants. Nonetheless, if you were to turn it around as a prediction problem, it does limit its approach. And in addition to that, it also limits the ability to identify the variant that is causally relevant or driving the association signal. So that caveat still exists, though, with these multivariate models. However, in the multivariate model setting, what you're trying to do is say, I'm going to fit a regression for my response, be it a continuous response or a binary response. And I'm going to fit it with the covariates in addition to all million variants that you have access to. So one of the challenges with these multivariate models is that typically the estimates of effect sizes or coefficients that you get at the end are less interpretable. They do introduce high computational costs. You do need more data. And at the end of the day, what you are optimizing for is for better prediction. So you do get better prediction compared to these GWAS univariate models if you were to build a predictive model on top of these GWAS univariate models. So in terms of terminology I'm going to introduce here, for UK Biobank, given that we have hundreds of thousands of samples that we're analyzing it, we refer to it as large scale. So that's very big N. Because we have hundreds of thousands of variables or predictors, we refer to this problem as high dimensional. So very large P. Now, the models that we developed or algorithms that we developed is called Basel algorithm. It is implemented in SNPNet package. And what it does is allows you to run lasso regression, elastic net regression as well, these penalized regression multivariate models in these large scale and ultra high dimensional problems. This first set of papers that introduced SNP-net and basal algorithm was published last year in PLOS Genetics. Here, just to walk you through what these so-called lasso models allows you to do in these multivariate problems is highlighted on the plot on the left. So to walk you through it, there are 4 curves on this plot on the left. The black and the red curves correspond to lasso regression. On the x-axis 1, that refers to lambda index, or as we move through the sequence of the model that's been fit, the less the model gets penalized for including variables. On x-axis 2, we indicate the number of active variables in the problem. So that includes also the covariates, but also the set of genetic variants that are introduced in the final path or solution of the problem. And as you can see, for the final solution that we decide for this trait height, standing height, we selected over 40,000 variables, where the performance in the training kept on increasing, but then the validation, in the validation data, you can see the performance of the model plateauing. Now, the green and blue curves correspond to the relaxed lasso. The difference between the relaxed lasso and the lasso is that in the relaxed lasso, you only use the penalized regression for variable selection. You don't actually shrink the coefficients. And you can see the property of, by comparing the blue and the red curves, the property of shrinking and the effect that it has on the overall performance of the method. You could see that the performance of the lasso algorithm outperforms relaxed lasso for standing height, at least in the validation data. So on the y-axis is R squared. That's the metric that we're using in order to optimize our fit. So the final solution that we have for standing height has an R squared of 0.7. Now, SNP-net is available, so anybody, it's an R package. It supports Plink files, PGen files, specifically Plink2 files. And we're continuously working on optimizations of new methods, new models. And right now we're actually working on SNP-net 2.0 that has like about a 5 to 10-fold increase boost in performance relative to SNP-net 1. And that's to take advantage of the property of sparsity in the problem, especially as we start moving into whole exome and whole genome sequencing data. So just to give you a sense as to what families are supported in SNP-net, we have support for the Gaussian family for continuous response. We also have support for binary response. So the logistic family is also supported. And for Cox proportional hazards models, so multivariate Cox proportional hazards model with very large number of predictors, these so-called time to event models. Now, the time to event model for survival-like data has also been published in Biostatistics, and that work was led by Ru-Ling Li, who's a PhD student working with Rob Tibshirani, Trevor Hasty, and myself. And we also do support lasso and elastic net regularization right now in SNP-net package. Okay. Now, going back to the problem of, or the application of this work to the context of the genetics of biomarker study, one of the things that we did was to train polygenic risk score models or multivariate models in these data. We applied it to the 70% training, 10% validation, 20% test split for what we refer to as and what UK Biobank refers to as the white British European subset of the UK Biobank. We also applied it to additional test set populations, including the non-British Europeans, South Asian, East Asian, and African population of the UK Biobank. And we estimated the relative performance across these non-British populations relative to the British population. Here, this plot, what it highlights is the performance of the polygenic risk scores across a handful of biomarkers that we selected to highlight. So first, we'll focus on the bar plot with no fill in it. You can see that the variance explained for lipoprotein A is approximately 50% of the variance explained by a polygenic risk score that we trained on the white British subset of UK Biobank. Alkaline phosphatase and LDL, we explained about 20% of the variance. C-reactive protein, creatinine, approximately 10%, and alanine aminotransferase are about 8%. So here we selected a set of biomarkers that are representative of the different disease categories for these biomarkers: the renal biomarkers, the liver biomarkers, cardiovascular biomarkers. One of the other observations from this plot as well is the performance of the polygenic risk scores in the non-British populations. For example, in the non-British European component of the UK Biobank, we do see similar performance relative to the white British component of the UK Biobank. And you do see a marked drop-off as we move to South Asian, East Asian, and African population groups. And I'll give you some hints as to why that may be the case, in addition to those some explanations that we've seen from the literature as well. So this highlights, of course, the transferability and lack of transferability of some of the polygenic risk scores that are trained from these biomarkers in UK Biobank, and also the need for increasing representation of some of these non-British European population groups as well in genetic studies.
Can I ask a real quick question?
Yeah.
You might have said it, but I might have just missed it. Were these polygenic risk scores created using only white British, or were they sort of trained on the entire population?
Great question. Yeah, that's a great question. So we're developing methods that combine the training across all population groups jointly. For this paper, we only focused on the models and methods that focused on the white British, or the largest set of samples in the UK Biobank. A few things was of course because these are new methods that are being developed and still we're evaluating the performance. We also wanted to highlight how much of a difference there is by just focusing on the white British subset. But yeah, so I think it's the first models that do show the performance of training polygenic risk scores at all in these biomarkers, right? So getting at the point of getting like 20%, 50% is quite high relative to what's out there. But yeah, in terms of what is next, developing models jointly is clearly the next steps. Okay, so another way to represent this or visualize this is to show the variance explained relative to a white British subset. Now, there's a few things here that may hint as to why there's this huge drop-off in non-European population groups. For example, if we zoom in on the African population group and look at the biomarker bilirubin, total bilirubin and direct bilirubin, now the final solution in the white British subset highlights about a handful, if not dozens of genetic variants to be included in the polygenic risk scores model for bilirubin. You can see that the performance actually is comparable in the non-British European subset, the South Asian, the East Asian, there's a drop-off in the African set. So that's kind of good news. And I think one of the reasons why we are getting this dramatic drop-off in performance is because we're not pinpointing the culprit or causal variants. So as you do train the model in your population group whose LD structure is way different from that in other population groups, and you using the properties of tagging, even if you're using lasso elastic net regression, it's going to use the properties of tagging at the end in order to identify the variables to be selected in this set. So one of the limitations is that then as you move it into those models into another population groups whose LD patterns are completely different, and you're not capturing the culprit variant that actually does influence the levels of the biomarkers, you drop off the performance of those polygenic risk scores. So one clear example of that is lipoprotein little a. You can see lipoprotein A has, we've shown that the performance is pretty comparable in white British and non-British European groups. But then as you move to South Asian, East Asians, and African, it drops off dramatically. Like we do explain about 50% of the variance using tags in the white British subset, non-British European subset. But as you move to African, that variance explained is approximately 0%. Now, one of the reasons that the conjecture that we put forward is that lipoprotein A has this VNTR of many amino acids, repeats a copy number variant that is pretty difficult to type. And a nice paper just came out a few days ago showing that they were able to call lipoprotein A in the UK Biobank and impute lipoprotein A copy number variant calls in UK Biobank as well, in some other samples, the independent samples. And they were able to show that they captured the relationship between the copy number variant and lipoprotein A. And actually in African samples, they do improve their ability to predict as well lipoprotein A levels. So one of the cool things that follows is that because we're training models that don't include the causal variant, as we transfer it to other populations where that LD pattern just doesn't match, the performance drop-off is quite dramatic. Whereas in bilirubin, that's not the case, where we actually do have the set of causal variants in the predictive models because it only chooses a dozen or so variants, and then transferring it to the population actually has pretty sustainable performance. Now, that still doesn't introduce the other limitations that may include from other population groups, including sets of genetic variants that influence variation of disease predisposition or biomarker levels in that population, variants that might be at higher frequency that will just be underpowered in European population groups. And in addition to that, I think there may possibly be some genetic variants that do have heterogeneous effects across population groups, but that still seems to be a bit less of an issue. I think that the major issue right now is making sure that you're identifying the causal variants and making sure that you're considering the LD reference, the LD references, even if you can't get at the causal variants, make sure that you have the LD properly configured to tag the causal variants. And that's a hard problem in these other population groups. And of course, the last one is in these other population groups, making sure that you capture the missing heritability that could be largely explained by population groups that just is not explained, that just doesn't contribute much in the commonly studied population groups right now. So I think this is an exciting area of research, and I think the more and more we combine some of these data, build models, taking these things into account, it becomes clear as to how we can reduce some of those differences in the performance of the polygenic risk scores. So with that, one of the questions we wanted to ask by using the polygenic risk score is to see whether or not they had any disease relevance. So one of the things that we did with the UK Biobank held out data was to ask the question, given the polygenic risk score predictions, are there any diseases that are associated in a FIWA style for these biomarker polygenic risk scores? So what we did was to compute the odds ratio of a disease outcome if you were at the 1% tail of polygenic risk score or the 5% tail of polygenic risk score, and compared that to a 45 to 55% bin, and applied the FIWAs to approximately 100 disease outcomes. For example, for the renal biomarkers, you do identify renal kidney failure, gout, for example, for urate. You identify diseases that are biomarker, that have relevance to biomarker diabetes with HbA1c. So then one of the ideas that came about from thinking about this problem was that where there's a few observations that we've made and that have been made in earlier studies as well. So we're definitely not the first ones. So endophenotypes are definitely more heritable than diseases. Here's just an example as for risk factors for coronary artery disease. Here, if we were to indicate the heritability of coronary artery disease as a disease outcome, the heritability is about 0.4. For A1C, Lp(a), BMI, LDL, risk factors that we know about, the heritability is higher, around 0.75, 0.79, 0.94, and 0.6. Now, so you can ask the same question, is the same true of the polygenic risk score? So if we were to come up with an idea of combining the polygenic risk scores for the risk factors, possibly the endophenotypes or biomarkers that may be associated with it, can we improve our ability to predict a disease outcome beyond what a polygenic risk score for that disease provides? So we tested this out with multiple diseases. This sort of quote unquote, or what we refer to as multi-PRS models, do improve prediction of prevalent kidney disease risk. Here's just an example of chronic kidney disease. The gray line refers to the polygenic risk score trained for chronic kidney disease using the disease outcome alone. And then the red line refers to the performance of this multi-PRS, where we combine the biomarker PRS in addition to the disease outcome PRS to build a multi-PRS model for the disease. And you can see that there is some predictive, improved predictive performance for chronic kidney disease prevalence across the polygenic risk score quantiles. Then on the right-hand side is just representing the data in terms of area under the curve. So what we did was to then evaluate the performance of these multi-PRS models relative to a PRS model for the disease itself with the held out UK Biobank participants. You can see for diseases like chronic kidney disease, cirrhosis, gout, and type 2 diabetes, you do see a improvement in predictive performance. We asked, are the findings from the UK Biobank transferable to an independent population cohort like FinnGen? Here, again, the limitation is that it is a European population cohort. And what we see is that the results are consistent as well. So we were to use the PRS from the disease outcome and compare that relative to a multi-PRS, we do see an improvement in performance for diseases like gout, cirrhosis, chronic kidney disease, and type 2 diabetes. Nonetheless, for diseases like myocardial infarction, that was not the case, even though it was the motivating example. And I think one of the reasons, and we include this in the discussion as well, is that myocardial infarction right now, to date, has, at least when the study was conducted, has such a great PRS that it probably captures most of the variation that is needed to be explained by a PRS score. Whereas for other diseases where it's still in the early stages of some of the disease studies, it may well come a time when the disease PRS gets so strong that you don't need the biomarkers in addition. Okay, so with that, one of the I did want to highlight a bit some of the work that we did with testosterone levels. Now, for all the biomarker levels, one of the things and one of the patterns that we tried to assess whether they existed was are there any sex-specific effects? For most traits, the genetics between the genetic effects between males and females is pretty concordant. There are a few exceptions where there is this discrepancy between genetic effects that exist in males and females. And testosterone level as a biomarker was one of those where the genetic effects were just completely discordant. Here, what we identified was that here in this plot, at least what I show is the estimated effect of genetic variants that we've identified to be associated with testosterone levels estimated in females. And on the y-axis is the estimated effect of those genetic variants that we've identified to be associated with testosterone levels in males. And you can see that the effects are quite discordant from each other. It pretty much looks like disjoint set of genetic variants or biology that affects testosterone levels in males and females. This work was led by Emily Flynn, who's a PhD student also in the biomedical informatics program at Stanford. Now, one of the interesting ideas that came out of this as well was this concept that, this may be an opportunity where you train polygenic risk score models independently in males and females, and that may boost your performance in prediction of the biomarker levels relative to just training a model jointly using males and female data. Here is the demonstration of that. For example, the plot on the left-hand side, the orange dot and whisker plot corresponds to the polygenic risk score performance in male testosterone if we were to use the female-specific genetic variants. It's pretty much non-predictive. When you use the combined polygenic risk scores to predict testosterone level in males, you do get improved predictive performance. And now if you use male-specific polygenic risk score models to predict testosterone in males, you do get a much bigger boost in testosterone prediction levels in males. And the similar pattern is identified in females as well. Now, the market drop-off is, of course, because of this pattern that the genetics of testosterone levels in males and females is just not overlapping. So we use the sex-specific instruments to assess causality using Mendelian randomization analysis. And one of the things that we'd like to highlight here, at least, is the pattern that if you were to use male-specific variants as instruments in a Mendelian randomization analysis, you identify that there's this strong protective effect against type 2 diabetes in males, and no effect for either the female-specific variants or the male-specific variants as instruments in females. So for anybody who's interested, we did publish this study in the European Journal of Human Genetics, and the polygenic risk scores are available on Figshare for anybody to use. So as far as key takeaway for this study, there are multiple rare coding alleles that we've identified with large effects associated with serum and urine biomarker levels. The second point I think is important, but I think that, given that it's in silico, it does have limitation. There's still a moment in time where I think for fine mapping to be completely convinced, you do have to do functional follow-up. And, the same thing with the rare coding alleles, but it's easier to break the LD patterns with rare coding alleles. Whereas with common variant association is a bit more difficult, even though these in silico methods suggest that they are the culprit variants. Third, these multi-PRS outperform existing PRS models for some diseases. And for testosterone biomarker level, at least, the sex stratified testosterone prediction levels did improve ability to predict testosterone levels in males and females separately better than by combining. We've made the polygenic risk score weights, associations, and the FiMAP data available in Figshare for anybody to use. So feel free to download some of those results and compared to some of the results that you may get as well from the data. With that, I'd like to thank all the members of the lab who've been working on these projects and who have made these findings possible. Thank you.
Excellent.
Thank you, Manuel. I was just going to jump in with a question, if that's
Go
okay.
ahead, Nick.
Okay, cool.
Yep.
Thank you so much. This is Nick Timetti. Follow your work. It's awesome. And so I'm really excited you're here today. And thank you for the great presentation. So I just had a question. I just to get your thoughts and a little bit of vision from you. So as we look at 250,000 exomes that are out, or 200,000 exomes that are out now and more coming out, and then whole genome, which is the first release is going to be by the end of the year, I heard. Can you talk a little bit about how to translate some of the stuff you've been doing with genotyping over to that, and the different types of analysis that will enable, what you're looking forward to?
Yeah.
And then just as a follow-up, because I think it's especially exciting to address, that those data will help to address limitations when the models are applied, different ethnic groups and ancestry groups.
Yep.
So could you talk a little bit about that too?
Yeah, absolutely. So the exome and whole genome sequencing data, so I'm excited about the exome sequencing data just because of the depth of new genetic variants that are going to be identified. In the array design, I think there were tens of thousands of genetic variants that were included. Pretty comprehensive from a coding point of view, but it does hit a limitation of like about 1 in 10,000 frequency. So, if you're looking at these like ultra rare signals, I think they'll be very exciting. One of the other cool things about the exome sequencing data, I think, that Poru just showed proof of principle, that they were able to do structural variant calling and then do the imputation of some of the VNTRs into UK Biobank and highlight, new genetic variants that are there but weren't discovered or typed properly that explain a large fraction of the variants for some of these traits. But I think in terms of some of the lessons here that may still translate over to exome sequencing data, we're still trying to figure out how much new associations emerge from the exome sequencing data relative to the array and the imputation data. I think one of the cool things will be in order to map out the allelic series that exists for any given gene. So, you may know that ApoB genes is affecting ApoB levels, right? Of course. So then the cool thing then is to figure out from just, experiments of nature, just humans that carry mutations, how is it that mutations in ApoB, the allelic series ends up playing out? And there you could start to see patterns of, some mutations that increase ApoB levels, some mutations that decrease ApoB levels. And the hope is that then you'll start connecting it to the function of that protein. And, whether there'll be enough saturation, it's unclear. But I think right now, at least for the early signals that emerge from gene hunting expeditions, I think that's one of the cool opportunities that's emerging from that. For whole genome sequencing data, there's still a lot to learn from the non-coding space. So the question there is, will we ever be able to constrain the space of assessing associations for non-coding genome to a point that will give you important units that are analogous to exomes, right? Like gene-based units. And I think that the work from folks trying to figure out like what the rest of the genome does is clearly important. Like why is it that some of these regions of the genome are more conserved than others, and are they active in specific cell types that are clearly important in renal and the liver space. And I think that's where the biomarkers come in handy. And the biomarkers come in handy because you know that you're getting 1 unit closer to the protein, but still not the protein itself. I mean, for ApoB, it is the protein itself, but for like LDL and for other ones, they're not the protein itself. So you are still getting the polygenic nature. And I think some of the stuff that Jonathan is also working out, Pritchard. But I think in terms of learning about like what the non-coding genome does, especially as we move into rare variants, is going to be interesting. For whole genome sequencing data as well, I think one of the cool things that's going to happen is when we built out these predictive models, having the information about all possible genetic variants in one place will finally be cool to see. So I think that's one of the promising things about the whole genome sequencing data is like, if you were to build out these multivariate models, knowing all the genetic variant that exists, including like LP little a repeats, knowing all the possible copy number variants that could be called, could we identify the causal variants in one go, or do we need to build out these models that do it like a two-step or three-step process? So I think from a statistical point of view, it'll be interesting as well. One of the challenges that exists from a statistical and computational point of view is like how you represent all that information in a file format that is easily manageable, right? So like the exome sequencing data, I think when we downloaded it, it was approximately like 800 gigs, right? And the Plink file. Plink 2 now has this new file format, PGen, that takes advantage of the sparsity, and it compresses it to like about 16 gigs. Right, which is awesome. And I think the same opportunity exists with the whole genome sequencing data once we're confident with the calls. So the models that we're trying to build, so SNPnet 2.0 is basically to take advantage of the idea, some of the sparsity that's going to exist. So at the end of the day, we want to be able to fit the entire model in memory, and we want to be able to take advantage of the possible groupings that may exist, for example, by genes or by non-coding units that somebody ends up coming up with, right? For your second question about polygenic risk scores, it's quite clear that one of the things that needs to be worked on is on making sure that either it becomes, the transfer, I think the idea of transferability of polygenic risk score has limitations. And what we do need to do probably at the end of the day is probably to do joint learning of polygenic risk scores across population groups. I think the UK Biobank affords that opportunity, but it'll be great to see other cohorts. Especially in the admixed setting, it's clearly going to be challenging. So what do you do in that setting? You do need to, my impression is that you will need to learn about the haplotype of each individual independently as you move into that mixed setting. Region specifically, right? Like it's just saying somebody is from a population group is not really going to work out that well if we're looking in admixed samples.
Thank you.
Yeah, all right.
Great. Well, thank you so much, Dr. Rivas. This was great, and I think a great discussion at the end. I think we're just about out of time. Again, thank you so much for your presentation. And
Thank you. All right, thank you. Thank you very much, and I look forward to seeing you in the future.
All right,
Bye-bye.
thanks everyone.
Bye-bye.
Bye.